So good morning to everyone. Welcome to Enjoy Science. It's Wednesday morning, so it's again time for the large family of MD Andersonians from inside and outside the institution and from all over the world to come all together to listen wonderful talks from wonderful scientists, scientists and clinicians. Because this is what we love more. We love to do science, we love to do outstanding clinical work, we love to do with our patients, and we love to be with all of you who are from our family. And because this is the last session of the year, let me and all the people who produce Enjoy Science to wish you happy holidays and to wish that 2021 will be a better year for all of us from a lot of points of view. Of course, Enjoy Science will move forward at full speed, so we will continue at least for the first part of 2021. And I have to thanks, of course, to Giulio Draeta, who accepted my idea some time ago in March, April to start this Enjoy Science. I have to thanks to Ellison Gard, Liz Kent, and Laura Adame, because without their wonderful work behind the scene, nothing can be done. And of course, I have to thanks to all of you who attend really hundreds and hundreds each talk, who are the attendees. Without you, who listen, who ask, who understand what we are doing and the speakers are doing, no webinar can take place. So thank you again, all of you. And with this, let me to thanks very much to Dr. Lauren Byers to be with us. Why? Because she is very busy, because she loves what she is doing. She is a wonderful clinician scientist, and this, especially this time of pandemics, it's a very tough time for all of them. So let me to tell to all the clinicians and clinician scientists from MD Anderson and all over the world, thank you for what you are doing for everybody during such a difficult time. And with this, many of you, most of you, we know Lauren. We know she's a bright and excellent clinician scientist. She is now an associate professor in thoracic head and neck medical oncology. And basically, she has an extraordinary training. If I remember well, she is trained in Princeton, Baylor, UT, and Johns Hopkins. This is best of the best. Why? Because she is among best of the best. Let's be fair. In this country, big university do not select everybody. Select who are the best ones because they represent the names of this institution exactly as we, most of us who attend, we represent the name of MD Anderson. Of course, she gets a lot of accolades, and I want only to say that she was a Sabin Fellow, which is a very important distinction inside the institution, and also other wonderful prizes. And she's publishing extraordinary work, extraordinary collaborative work with a lot of clinical translation in what is the biggest killers among all cancers, lung cancer. And with this said, thank you, Lauren, for being with us, and please take the show and good luck with your presentation. We are all very curious about your very interesting data. The title, it's very, very interesting and make us very curious. Good luck.
Thank you. Well, thank you so much. And I really appreciate the chance to be here and the invitation to speak. And I'm excited to share some of the more recent work that we've been doing in small cell lung cancer. And so today I'm going to focus on work that we've been doing with biomarkers, but also in particular how we've been using liquid biopsies and blood samples from our patients to really dissect, what's going on in this cancer and figure out a path forward. These are my disclosures. I think that we have all been just incredibly fortunate to be part of cancer research during a time where we have seen amazing transformations of cancer care. And this has really, been driven through scientific breakthroughs. So when I was a medical student, I had the chance to work at the NCI during the early days of molecular profiling, and actually got to go, again, as a medical student to the party where they celebrated completing the first human genome project. And so this was back in 2001. That was also when the first targeted therapy was approved for cancer treatment and leukemia. And then very rapidly, I think, looking back at this timeline, 10 years later, we have the, go from the first targeted therapy approved to the first immunotherapy approved. And we've just seen, an explosion of research and understanding of ways to harness the immune response for an increasing number of patients, increasing number of cancer types, to really get more effective therapies for many patients and much more durable therapies for our patients. I just want to take a minute to also recognize, I think we are literally in the midst of another historical moment for science and translational science and what people can do when they come together. And so I was just, yesterday looking back at the timeline for the vaccine for the SARS-CoV-2 vaccinations. And so in July, the phase 3 trial of the Pfizer vaccine started. In November, they reported out interim analysis showing success of the vaccine. And on December 8th, we saw the very first person shown here in this picture, the screenshot from the New York Times. This woman was the first person outside of a clinical trial to receive the SARS-CoV-2 vaccination over in the UK. And then very rapidly have been fortunate to have this vaccination, and hopefully the Moderna vaccine coming soon as well, approved in the United States with a vaccine arriving on the 14th at MD Anderson. I also, before I start on my topic on liquid biopsies, I just want to give a shout out to my lab. So, I think researchers, clinicians, investigators around the world really have teamed up and have contributed in whatever way they could, both from patient care through research aspects. And so during the lab shutdown, when our research labs at MD Anderson closed on April 15th, we all came in on a Sunday and shut everything down and got everything ready for an unknown period of time to be out of the lab. And we were out until June 11th. But during that time, my research group, both the scientists in my lab, as well as our incredible bioinformatics team, we realized, here we have one of the world's largest databases of lung data, and it's lung cancer data, but lung cells. And so we realized that we could leverage that to try to ask questions that might be pertinent to the pandemic. And so we have a paper that we have published online and a revised version that has been submitted recently, where we again leveraged our lung cancer data to really try to understand how the ACE2 receptor that's used by the virus to enter cells is regulated, and we've identified a potential therapeutic target that might be relevant for treating patients with COVID-19. My interest, I mentioned that I was fortunate to be at the NCI. I was a Howard Hughes Fellow during medical school and spent a year there, and again was really lucky to land in Lou Stout's lab. So Dr. Stout, who had become the head of the TCGA more recently in his work, but at that time was doing pioneering research using a very new technology then, using RNA or cDNA microarrays to measure the RNA expression of about 8,000 genes using a microarray that they called the lymphochip. So this was, again, back in 2000. So this is a picture of me as a medical student, and I was part of what was called the Lymphoma and Leukemia Molecular Profiling Project. And this was a study where we were looking at diffuse large B-cell lymphoma at the mRNA level to try to understand why some patients with standard treatment at that time would go on to durable responses to treatment, whereas other patients had early relapse that was then very difficult to treat. And what we found as part of that work was that even though these cancers looked very similar, under the microscope in terms of the histology, there were clear patterns that emerged in terms of different genes that were activated in different patients' cancers, and that really predicted which patients were going to do well and respond to standard treatment, and which patients had more aggressive and less sensitive disease. And so this led to a landmark New England Journal of Medicine paper that I, again, was fortunate to be part of this team and to be able to have this experience. And I think this really sort of set up my interest in using molecular profiling and bringing this to solid tumors, and in particular lung cancer, and how we could take similar approaches to define new groups of lung cancer based on their molecular profiles, as well as biomarkers that might help us to move forward with more personalized approaches to patients. This is from an editorial that I wrote a couple years ago, but I think this kind of frames, the approach that my lab has had to a lot of our work, and that is that we want to leverage these incredible, high throughput, big data approaches that have really, become available to us as cancer researchers. Use that, and this is very a close collaboration that I've had for more than 10 years now with our bioinformatics group, including Jing Wang, who leads that group, and go from the big data to what are the most promising candidate drug targets or biomarkers, and then get those into clinical trials as quickly as possible. And then what we learn from the clinical trials, we then take back to the lab, and then figure out the next steps forward. And as you heard, my lab has really focused a lot on small cell lung cancer. And so this is a type of lung cancer. It makes up about 13% of newly diagnosed lung cancer patients each year in the United States. But unfortunately, unlike non-small cell lung cancer, where now patients who present with advanced disease, the first thing that we do is we're getting mutation testing, PD-L1 levels to assess, if the patient's likely to respond well to immunotherapy. We have a growing number of biomarkers that help match patients to personalized treatment approaches. In contrast, in small cell lung cancer, at the moment, there still are no approved targeted therapies, and there are no clinically validated biomarkers yet, although we're working hard on changing that. But I think one of the challenges of this disease is that essentially, the treatment still is very much kind of a one-size-fits-all. And it's a very aggressive cancer. It does tend to respond initially to chemotherapy and to radiation, but then rapidly, usually within a few months of that initial treatment, relapses and then is highly resistant to most existing agents. So let me see if I can get the pointer here. So the approach that we've taken is really to build a resource of molecular profiling data across our preclinical models. And so these are the cell lines. We have almost 300 profiled cell lines from both small cell lung cancer and non-small cell lung cancer. We have, several different mouse models now, both genetic models as well as patient-derived models that we can use, as well as cohorts of patient tumors. And what we want to do is really understand, what are the DNA profiles, the RNA profiles, the proteomics of these, and more recently have started also looking at the single cell level, which is really helping us to understand heterogeneity and how having multiple different sort of cancer cell groups within an individual tumor may contribute to why these cancers are so resistant. The other thing, and this will be a big focus of what I talk about today, is how can we also leverage blood samples from patients in order to study this cancer. And this is important for a couple of reasons. One is that small cell lung cancer, unlike many other types of solid tumors and unlike non-small cell lung cancer, is for the vast majority of patients is not treated with surgical resection. And so that means that for the most part, when we have tissue samples, they're small biopsies. This is the reason that we don't have a TCGA of small cell lung cancer, because there just aren't, banked specimens with surgical resections for these patients to allow us to do that. But because these cancers are so aggressive and metastasize early, there are a large number of circulating tumor cells in the blood, as well as quite a bit of circulating tumor DNA. And so this provides an opportunity to use blood-based samples for studying the disease, and also for being able to follow over time, how is it changing, how is it adapting, because what we really want to understand is, what is driving the resistance that we see emerge and a very different behavior after a patient has received their initial therapy. So the three main areas that we have focused on in order to try to make more progress in terms of using translational research to get more effective treatments to the clinic and to understand the biology of this cancer better is, as with other types of cancers, we recognize that small cell lung cancer truly is not one disease, even though the current standard treatments, are basically similar for different patients. And so, our group and many others in the field have been doing a lot of work to define what are the subtypes of small cell lung cancer. And unlike non-small cell lung cancer, what we have seen is that these are not going to be mutation-based markers. So it's not going to be, for example, the equivalent of an EGFR mutation or an ALK fusion, but rather these are based on differences in expression of RNA or proteins between different small cell lung cancers. And so we want to define the subtypes that'll let us move forward with candidate biomarkers and approaches to move towards more personalized patient treatments where they're being matched to specific therapies based on their profile. We also want to address this challenge in terms of the limitations of tumor tissue for translational research. And so in addition to the fact that patients typically don't undergo surgical resection because, we know that the recurrence rates are so high with this cancer, frequently recurrence is diagnosed based on imaging alone. Patient gets a follow-up surveillance CT scan or PET CT scan, and there's, new mass or new areas. And so frequently in standard practice, because we want to get patients started on their next treatment quickly, they have not frequently undergone repeat biopsy. And so, in the context especially of the clinical trials that we're doing, we've been working very hard to build in biopsies, preferably before and after treatment, so that we can really start to profile not just treatment-naive cancer from original diagnostic biopsies, but also having samples taken after relapse so we can understand more about resistant disease, and then beyond that so that we can understand how the cancer is evolving and mechanisms of resistance to other new treatments that are being tested. Related to that, again, as I mentioned, in addition to the biopsies, we've set up a translational platform in thoracic medical oncology that allows us to offer patients the chance to participate in translational research by consenting to having blood collected. If they're undergoing a biopsy as part of their standard care, having additional tissue collected, and using these blood or tumor samples to make new models that we can then study in the lab to better understand emerging treatments and emerging biomarkers. And again, the final point being the fact that these cancers evolve very rapidly. So we know that, as I was saying, when we first treat patients, the majority of patients respond to their initial frontline therapy, which for most patients is a combination of chemotherapy and immunotherapy. But about half of patients will have the cancer relapse within 6 months. And at that point, responses to other existing therapies tend to be very low, so around 10%. And so we really need to figure out what has changed between before the cancer saw chemotherapy and immunotherapy to afterwards, and how can we use that information to guide us to more effective therapeutic approaches. One of the first real, I think breakthroughs that we had in this was actually when I was a medical oncology fellow, and I was working closely with John Hamack and Gordon Mills, who were two incredible mentors. And we started by asking, using our proteomic platform, if there were differences in the different signaling pathways or proteomic profiles of small cell lung cancer as compared to non-small cell lung cancer. And one of the major observations that emerged from that was that we saw this very clear signal that genes and proteins involved with DNA repair response, such as PARP1, CHEK, ATR, ATM, were very highly expressed in small cell lung cancer as compared to non-small cell lung cancers. And so I'm showing this here. This is IHC for PARP1 in small cell lung cancer, and then another high-grade neuroendocrine cancer that's similar to small cell called large cell neuroendocrine cancer, whereas most of the squamous cell carcinomas and adenocarcinomas had much lower expression levels. And so PARP was a really interesting target for us because at that time there were clinical trials of PARP inhibitors that were in very late stage of clinical trial development, so particularly in BRCA mutated cancers. And so we knew if we could see activity in preclinical models of small cell lung cancer, there would be the potential to move this forward very quickly for our patients in the context of a clinical trial. And so we were fortunate to do that. We looked at multiple PARP inhibitors, and we saw that they had activity in a subset of the cell lines, as well as in mouse models. And so this led to two early clinical trials testing PARP inhibitors for the first time in small cell lung cancer. And this, the CT scan on the right shows the first patient that we ever treated with single agent talazoparib. This was the first patient to go on and had a follow-up scan 4 weeks later that showed that in multiple metastatic sites there had been a very significant decrease in the size of the cancer. We also saw that, while there were patients who were responding and appeared to be getting benefit, that there were other patients that were not responding to therapy. And so we went back and used our proteomics and other profiling data that we had from our preclinical models to start looking at what would be potential biomarkers for response to PARP inhibitors in small cell lung cancer. And we expected that this was going to be different than what had been described for other cancer types, because we knew that, for example, BRCA mutations and other alterations in DNA repair were extremely rare in small cell lung cancer. So that was not what was explaining the responses that we saw either in the lab or in patients. But our group and a couple other investigators working in small cell lung cancer found that in the small cell lung cancer models, the top biomarker that was emerging from the cell lines was a gene called Schleffin-11. So this was a gene I had never heard of before, I knew nothing about, but Schleffin-11 is what causes cancer cells to undergo cell cycle arrest and then apoptosis when there is DNA damage present. So if this Schleffin-11 is present and you treat with a PARP inhibitor that causes DNA damage, then the cells that have Schleffin-11 are going to be killed following treatment with a PARP inhibitor. We were we saw that initially in the cell lines, and then we were able to work together with some collaborators to run a co-clinical trial looking at this is talazoparib in several PDX models of small cell lung cancer. And so we basically grouped the different mouse models into whether that model responded to treatment, if it caused some stabilization of disease, or if essentially it did no better than the vehicle. And so when we then compared again the biomarkers between animal models that had some response to therapy versus those that didn't, in the animal models similar to the cell lines, the Schlefin-11 gene came out as the top predictor. So it was high in all but one of the models that showed response to the PARP inhibitor. So we were able to then work with Ignacio Westuba's group and Junya Fujimoto using our proteomics and our other gene expression profiling data to optimize an IHC. I'm showing here the IHC from the mouse tumors, but this really enabled to move this biomarker forward into the clinical trials that were ongoing at the time that we were doing in patients. And what we found was, so first of all, this is, the kind of biomarker that you would like to find. Half of small cell lung cancer patients express it. So 50% of patients have the biomarker. And essentially, it's kind of binary. Either tumors express it or they don't. And so we were able to essentially group patients into, whether or not they had any expression at all or no expression. So if you think about, this is kind of like what we use now in immunotherapy, thinking about, PD-L1 and is there PD-L1 positivity. And in this case, when we went back and looked retrospectively at patients who had been treated on the first randomized study of a PARP inhibitor, these were patients with relapsed small cell lung cancer who received either chemotherapy, temozolomide, the TMZ, with placebo, or temozolomide with veliparib. The patients who had the addition of veliparib and who were positive for this biomarker had significantly longer PFS and overall survival. So this is showing the overall survival data. The patients in the PARP inhibitor combination group that were biomarker positive had an overall survival of around 12 months, which is very long, especially this was primarily third-line chemo-refractory patients. And patients that did not have the biomarker, the overall survival was about 7.5 months. So I think this was, it was a retrospective analysis, but very encouraging, an encouraging signal to suggest that we should continue to follow this up further. So one of the challenges with looking at Schlefin-11 or any other biomarker in small cell lung cancer is that, in the trial I just showed, we were using archival tissue. So basically whatever tissue patients had available from their original biopsies from their original diagnosis. And even though we enrolled 100 patients onto that trial, only half of those patients had enough archival tissue from their biopsies to do any biomarker testing. And so again, it's a major challenge in terms of having enough tissue to be able to do the biomarkers in the first place. And so we were interested in, again, can we leverage blood for detection of biomarkers of interest? And we know that, in other types of lung cancer that there can be a lot of variability in terms of how much, how many CPCs are in the circulation or how much ctDNA may be in the circulation. So that can be limiting and can lead to false negatives where you might not detect something in the blood, but you might be able to detect it in the tumor itself. But in small cell lung cancer, because there are so many CTCs and the amount of ctDNA in the circulation is so high, it really is sort of the ideal type of cancer to use these types of technology. And so this is my how I think about it. So that a lot of whether or not you can use blood-based markers depends on how much DNA or CTCs a cancer sheds. And in the case of small cell lung cancer, it sheds quite a bit. So again, the opportunity here with small cell was that we knew in a blood draw that there would be much higher numbers of circulating tumor cells compared to other cancer types. Also, patients were very, have been very willing and enthusiastic about participating in research and contributing to, moving the field forward and being partners with us. And so this, this also provides the opportunity for, longitudinal samples. So you can get one before they start treatment. You can draw blood again later on when they're coming back in for follow-up. So minimally invasive. And in the circulating tumor cells, we've been able to start building out together with collaborators biomarkers that are, of interest to us for this particular cancer. And so in the top right, you can see an assay that we've worked with Epic Sciences on to develop to look at Schleffin-11 directly in the circulating tumor cells. And so, so we can detect whether or not CTCs are present and whether or not they are expressing Schleffin-11 from a tube of blood. Across the bottom, it shows each column is an individual patient sample that was sent for analysis. And what you can see is that, essentially all of the patients here had detectable circulating tumor cells, but then there was variability in terms of which patients had CTCs that expressed Schleffin-11 or not. And often within an individual patient, there were Schleffin positive and Schleffin negative, so biomarker positive and biomarker negative cancer cells. And that's important in terms of being able to capture not just if the biomarker is present, but is it a mix of marker positive and marker negative cells? Because we really, again, are trying to understand how the cancer is changing over time, and we need to understand, is there greater heterogeneity? Is there sort of a greater, mix of marker positive and marker negative cancers as you go further along in treatment? And that may be what is driving resistance to, many of the therapeutic drugs that we're currently, either have been tried or are being investigated. So just going back, this was an initial pilot study where we were looking at blood from patients who either had not yet received any treatment for their lung cancer or who were on treatment or who had recurrence of their cancer. And in the circulating tumor cells, similar to when we look directly at the tumors, we know that there about half of patients have expression of Schleffin-11, and that was true looking at the circulating tumor cells. We've since profiled a much larger number of patients at this point using the CTC analysis. We still see the same thing holding, where about half of the patients have expression of the marker at their initial diagnosis. But then if they are on treatment or if the cancer has relapsed, then we see the expression of this marker go down quite a bit. And that is consistent with what our group and other investigators have described in their laboratory studies, that if you treat, for example, with cisplatin chemotherapy in a model that is expressing Schleffin-11, we very quickly see that it is downregulated. And so we think that the downregulation of this is an adaptive mechanism of resistance to DNA damaging therapies. So the other thing that we were interested in doing with the circulating tumor cells, and this was really driven by Allison Stewart, who's an incredibly talented staff scientist in my group, and she had read a landmark paper by Carolyn Dives' group in the UK that had shown that you could take blood samples from patients, deplete the lymphocyte population, put them into the flank of a mouse, and after some number of months, essentially grow a PDX just from the blood sample and the CTCs that were present in that blood sample. And so Allison was very interested in doing this and partnered closely with Carl Gay, who's a physician scientist in our department. And the two of them started, collecting blood samples and developing these new circulating tumor cell derived mouse models. And so again, they would put the CTCs into the mouse, and then once the tumor had established in the mouse, it can be expanded into additional mice, and that gives the opportunity to do, first of all, the same type of molecular profiling on the tumors that we've been doing with our other models, so we can know, what are the genomics of this. We call them CDXs, CTC-derived xenografts. So what are the genomics? What are the proteomics? What's the gene expression profile? And then we can also, expand the mice and test with either standard treatments or novel therapeutics that we're investigating. And this shows from a paper that we published at the beginning of this year in Nature Cancer, some of the first models that we made from patients who participated in this research program. And so we had, as an example, we had 4 patients that were chemotherapy sensitive. So either they had not yet received any chemotherapy or they had received a very minimal amount. And then we had 4 patients who were chemotherapy refractory or had acquired resistance. So patients who had received chemotherapy and then later relapsed, or in one case, patient who never responded to chemotherapy, which occurs in about 20% of patients who are newly diagnosed. And so the red triangle shows you where we collected blood. So what, the time points at which we collected blood and the CDX model was established. And again, I mean, this is using a single tube of blood. Sometimes it can be anywhere from 3 cc's up to 7 or 8 cc's, but a single tube of blood, and we have about a 35% take rate in Allison's hands for establishing a flank tumor. Sometimes they can take quite a while for them to establish, but then once they do, then often when they're going to additional mice, then they start growing more quickly. The other thing that was really interesting for us and unexpected was that these cancers, the tumors in the mouse models really maintain kind of the aggressive clinical behavior. And so we have now seen in several instances where even though the tumor, the cells are implanted into the flank of the mouse and grown, and passaged in the flanks, we've seen metastasis to distant sites. And so I'm showing on the right just an example of one of the mice who developed leptomeningeal disease, so metastatic spread to the lining of the brain. And this again was unexpected. It was really fortunate that this was a collaboration between the scientists in my lab and the physician scientists sort of on the translational side, because the mouse had developed some neurological symptoms that are not unlike what we observe sometimes in patients who have brain metastases. And so Dr. Gay had thought, well, maybe it's possible that the patient, that the mouse has developed brain metastasis. And so working with John DeGroot and his group, were able to start studying the what was going on in terms of metastatic sites, especially within the CNS. So I'll just go quickly through a few other kind of key pieces. One is that these CDX models express the same typical markers that we see in the patient tumor. So if we compare to the patient's biopsies, we see standard high-grade neuroendocrine markers, things like Ki-67, NCAM. The majority also express a lung-specific marker called TTF1. So we are confident when we have these new models that they are in fact small cell lung cancer. And as we would expect, and as others have shown, so Ben Drabkin and Anna Farrago, who have also led work developing these types of CDX models as well, they had described, and we've seen the same thing, that the sensitivity of the models to chemotherapy, for example, matches the response that was observed in the patient. So here's an example of one of the patients where we collected blood to establish a model after the patient had previously received chemotherapy and then had relapsed chemorefractory disease. And when we established that model and then tested cisplatin, which is really the gold standard in small cell lung cancer, the blue line are the animals that were treated with vehicle. The red lines are the ones who were treated with cisplatin, and you can see essentially there was no effect of the cisplatin, which was consistent again with the clinical, the clinical history from the patient. And we've seen that across, all of the models that we've tested so far. The models that have come from patients who were chemotherapy resistant do not respond, whereas the patients who came, the models that came from patients who were chemotherapy sensitive do respond to cisplatin. And so it gives us confidence in terms of being able to use these to get a sense of how active therapeutics, chemotherapies, and targeted therapies may be within specific patient populations or biomarker selected populations from these models. The other question that we have been really interested in, and this was something that I think with the emergence of single cell RNA-seq, we had an opportunity to look at this in more detail, is whether or not some of the just incredible resistance that we see develop in small cell lung cancers, if that was being driven by increasing intratumoral heterogeneity. And so we wanted to take a look at that, and we were fortunate to get funding through a pilot project to do some single cell analysis of these models, of these CTC-derived PDXs. And so to do this, we would harvest one of the tumors from the mouse and dissociate the tumor cells, and then we could sort out the human cells from the mouse cells. And so that left us with a pure tumor cell population of human cells. Now, one obvious limitation of this is it does not capture the immune microenvironment, since it is removing the mouse cells. But it was a good starting point for looking specifically at the cancer cell heterogeneity. And then once we had this kind of up and running, and we're seeing some really encouraging signals from this, we decided to pilot whether or not we could do single cell analysis directly from a blood sample and profile CTCs directly without having to put them through the mouse. And so I'll show some of that data at the end. And what we observed, and again, I don't think that this was surprising, but it was very consistent, is that if you compare across the top these 4 CDX models that came from chemotherapy-sensitive small cell lung cancer, they're more homogeneous and less diverse as compared to these 4 that are on the bottom, which are from patients whose cancers had developed chemotherapy resistance. And so, again, across the board, consistently, we see that within an individual tumor, there are multiple different kind of subgroups of cells that have different transcriptional programs activated. And when we went back and looked using various computational approaches at which gene pathways were activated and which things were higher in these individual subgroups, we repeatedly saw that in these relapsed small cell lung cancer, chemotherapy resistant tumors, that frequently we were seeing multiple mechanisms of resistance at the same time in some of these discrete subgroups. And so that may be part of the reason that going in with a sort of a single agent approach in the relapse setting may only be treating one particular subset of the cells or addressing one of the resistance mechanisms. And we've used a variety of computational approaches to look at this further. So assigning an intratumoral heterogeneity score, again, you can see that the platinum resistant models, this is much higher. And so, I think that this supports that even though we see these cancers shrink after the initial treatment, so on the, on PET scan, CT scan, we see, reduced size, but that when but that essentially you maintain heterogeneity, or you're promoting, new groups of cells and transcriptional programs to be turned on, and so you end up with this much more heterogeneous, much more difficult to treat cancer. And using the mouse models, taking one of the models that was platinum sensitive, if we treated it until the tumor developed acquired platinum resistance, we saw essentially the same thing. So again, just from a patient, from this model that came from a patient and was chemotherapy sensitive, if we treated with cisplatin, until relapse. We got basically the same thing that we had seen in the other patients who had relapsed chemo-refractory disease, where we saw, multiple different populations of cells, including ones that had undergone EMT transformation, which is a known mechanism of resistance in many cancer types, including lung cancer. And so, again, there's a lot more detail in the paper, but essentially what we've observed is that you start getting more heterogeneity, both in terms of the predictive biomarkers and the different therapeutic targets that are being expressed. And you also get, in many of the cases, a subset of cells that have undergone EMT that may also be driving resistance. And so, as one of my colleagues described at one time, you start out with something more akin to a farm, where you've got kind of a flock of sheep, or animals that are, these are the individual cells that are all like somewhat similar to each other, but over time it evolves into more of a zoo. So I mentioned before that when we had this working, doing the single cell RNA-seq analysis in our CDX tumors from the mice, we then asked the question, could you potentially do this directly in patients? And to my knowledge, this was the first example of doing it in a patient with a solid tumor. So this was one of my patients with extensive stage small cell lung cancer, and at the time, prior to starting any treatment, so basically, after the initial diagnosis, we were able to collect a blood sample. There were 84 circulating tumor cells in that blood collection that were profiled by single cell RNA-seq. She had a really robust response to therapy, and in fact, on the scan, essentially the cancer became undetectable. She had a complete response. And at that time, when we collected another blood sample, while she had a radiographic complete response, there was only one circulating tumor cell that was detected. But later on, in the course of her treatment, she did develop relapse and new metastatic sites. You can see over here new areas of cancer in the liver and other areas. And at that point, there was a significant increase in the number of circulating tumor cells in the blood collection from that time point. And consistent with what we had observed both in the CDX models from other patients as well as in this patient's own CDX model, the makeup of the CTCs at relapse was a much more heterogeneous and diverse population of CTCs compared to her initial blood collection. And so if you look here, when we first collected blood at diagnosis, and there were 84 CTCs, more than half of those untreated cells clustered together in cluster, what we would call cluster 1. But then at relapse, we saw large populations of cells with distinct expression profiles, and so much more complex cancer. And if you just map some these different clusters to some of the therapeutic targets that are in current use or clinical trials for small cell lung cancer, what you can see again is that there were not a lot of them expressed in the initial diagnostic circulating tumor cells. However, in the relapse setting where you have these multiple clusters, you can see that many of the targets are expressed, but they may only be expressed in a subset of the cell, so in one or two of the clusters. So, I think, in terms of where we are now, and this was an editorial that was written at the time that our paper came out, and also another paper from Caroline Dives' group, both of which illustrated the heterogeneity that was driving resistance in using these circulating tumor cells. I think we are very excited that we have these liquid biopsies where we can essentially take snapshots of the cancer at different time points and see what is changing over time, and then use that to understand how these cancers are escaping from therapy, what's driving resistance, and what are things that we could intervene on therapeutically. And so I think the CDX models really provide a new opportunity to address an unmet need in the field, to have models from relapsed lung cancer, and then also because these are such complex cancers, to then follow within an individual patient how they've changed over time. Okay, so I think the heterogeneity and the evolution and adaptation of lung cancers is really important. We know that this is a key driver of therapeutic resistance. But of course, as an oncologist, the question is, then what? So what do we do about this, and how can we intervene? One thing that I think we really believe will make a difference, and this has certainly been seen in other cancer types, is that we need to keep working towards moving our most effective therapies, and especially combination therapies, earlier into treatment. So we need to do the laboratory work to figure out what are the most promising therapeutic approaches, and then we need to try to get them earlier and earlier. And in particular for lung cancer, and we know that the patients that are having durable responses are patients where they have also received some sort of immunotherapy. So we need to also figure out how to more effectively engage the immune response. And so one approach that we are taking, so I mentioned at the beginning of the talk that PARP inhibitors were a new target we had identified for small cell lung cancer. Last year we published a paper that because of the type of DNA damage that the PARP inhibitors induce, it actually activates the innate immune response through this STING pathway. And so if you combine a PARP inhibitor with immune checkpoint blockade, we see these dramatic and durable responses to therapy. And so this is something that now is moving into clinical trials. So we have 2 clinical trials for our small cell lung cancer patients that are about to start, which will be testing these combinations. So we're really excited about that, and also building in additional biomarkers that we think will predict which patients may get the most benefit from these combinations. The other thing is, we clearly see that these cancers are evolving over time, but I think that we will be able to start using various approaches, including things like RNA velocity and other computational approaches, to predict where is the cancer heading. And there are probably a few predictable ways in which cancer evolves and the cells adapt and turn on new pathways in order to mediate their resistance. And so I think about this a little bit in terms of kind of similar to the when you're using Waze or Google Maps and all of a sudden there's a traffic accident or road construction or something, and the map reconfigures what your route is so that it can get you there as quickly as possible. I mean, this is similar to for those of us who live in Houston, if there's a big rainstorm, it's not uncommon that there's certain roads that flood and are blocked and you can't go that way. And so you kind of have your standard, alternate routes of which way to go. So I think we can start trying to use, various technologies to predict kind of where the cancer is heading, how it's kind of escaping the treatment, and preferentially target those vulnerabilities. And related to this, we know that that these cancers are a lot of the switching between different transcriptional programs is related to their epigenetic landscape, and the fact that they can by targeting sort of epigenetic programs, that we may be able to really shut off to a large extent some of this subtype switching or evolution that's going on and driving the heterogeneity and resistance. And similarly, the last thing that I'll say is I don't think we have to necessarily target every one of those different groups of cells have an effective therapy. We know that some of those subsets of cells, such as those that have undergone EMT, may be playing a key role in really driving the resistance. And so looking at, particular kind of Achilles heels within the tumors, and certain subsets that we can target may also be an effective way to think about, addressing this heterogeneity and resistance associated with it. So with that, I'm just going to thank the multiple people from my lab, from our incredible bioinformatics group. I especially want to recognize Allison Stewart and Carl Gay, who really have spearheaded establishing these CTC derived models and the approaches to them. My other many collaborators at MD Anderson, so John Hamek, Don Gibbons, Bonnie Glisson, Junio Fujimoto, Ignacio Estuba, and they are multiple sources of funding, which we are very grateful for. So with that, thank you so much. It's a pleasure to be here and have a chance to share this work with you.
Thank you, Lawrence. This was a wonderful talk, very, very interesting. And in fact, there are very many questions. So let's start with the question. So a question from a pathologist. How do you isolate individual cellular population for single cell analysis?
Sorry, can you repeat that? How do we?
Yeah, how do you isolate individual cellular population for single cell analysis?
How do we sort? How do we identify the population
of cells
for analysis?
The population during the single cell analysis.
Perfect. So basically we do, we use antibodies to select for human versus mouse and then for viable cells. And so that's what goes into the, into what we profile for the single cell analysis. When we're preparing the blood to go into the mouse to establish a new mouse model, we really use kind of a dirty prep where we're essentially just trying to deplete as many of the lymphocytes as possible, but we don't want to deplete any of the circulating tumor cells. So we have purposefully avoided any selection antibodies. So we're not selecting for the CTCs, but we're trying to basically weed out the other cell types as much as possible.
Thank you. Another question. Do you believe that PDX fully recapitulates the original tumor heterogeneity, even if they were derived from CTCs? How long do you think this consistency can last in the model?
Yeah, so that is a great question, and that's one that there's certainly a lot of questions that we still need to answer. We have observed that it seems like the patients who have liver metastases may have higher, the CTCs are more likely to turn into a CDX, and we don't know if that's something specific about, the liver metastases, or if patients with certain sites of cancer are more likely to establish these models. So I'll take a step back. So I think as we continue to build more and more models, we know what the heterogeneity is across, looking at biopsies or tumors that have been profiled. So we'll have a sense of whether or not we're seeing kind of the same sort of distribution. And so I think that's an ongoing question. The other thing, as far as how well is it sustained? So far we have seen that we've looked out across multiple passages of these CDX models, both doing genomics, proteomics, expression profiles, and they are very stable, and the heterogeneity is also seems to be relatively stable as well. But I think that's something that that we'll continue to be looking at as we build even more of these.
Thank you. Another question. Based on this heterogeneity, about the CTC and tumor heterogeneity, I think, do we have to resync all the negative single agent trials that have been done over the years?
that's a good question. So we, I think that there are two things. I think that we have recent work, which I didn't present today, defining 4 major subtypes of small cell lung cancer. So sort of akin to, ER/PR, HER2, breast cancer, for example. So I think that, first of all, we're going to have, moving forward, we're going to have a better sense of what are the major subtypes of small cell lung cancer. And I think that within many of the single agent negative clinical trials, that it's quite likely that if we look back, that for some of those clinical trials, they were highly effective, but just in kind of one of those main small cell subtypes. We just didn't know how to define those subtypes yet. But that being said, I think ultimately, yes, I think that as with, many other cancer types and leukemias and other things, that, combination therapy, and we do this up front for our patients, already, but I think finding the most effective combination therapies, and probably including, immunotherapy approaches as part of that, is going to be what gets us the most, durable responses long term.
Thank you very much. Now I take one more question because of the time. So for all the people who are asking, there are many others, please directly contact Dr. Byers by email. So because of time, we can take one more question. So can you comment on the differences from small cell lung cancer and small cell differentiation emerging in adenocarcinomas treated with targeted therapy, EGFR blockade, for example? Do they have similar features?
So that is a great question. So we do think, there are many cancer types now where transformation to small cell is becoming more and more common and a major mechanism of resistance. We do think that there are going to be kind of key shared biology in many of those cancer types, and probably in EGFR mutated lung cancer is really an important one that we're very interested in. We have a couple models now of that where we're starting to study it, but I think we have to. I think there's more more to come. Interestingly, just as I was mentioning, we see different types of small cell lung cancer. It looks like the EGFR mutant lung cancers that are transforming, may not all transform to the same type of small cell lung cancer. So I think that's something that, but I do think that the therapeutic approaches for EGFR transformed cancers, may be similar, and I think a lot of the things from small cell lung cancer will be able to bring into that space.
Thank you very much. And with this, let's thank you very much for the wonderful talk and for the time for Enjoy Science. And I want to thank to all of you who attend, to wish to all of you a wonderful, a much better 2021 and happy holidays. Enjoy Science will continue unforced for the full first half of 2021. And with this, I can tell you only enjoy science, because in fact, science is what brings the society forward. And thank you very much again, Dr. Byers, for the wonderful talk. And thank you all who are attending, and to all MD Anderson family. See you in start of January. Happy holidays to all of you, and happy 2021.
Thank you. Thank you so much. Great being here.